The broadcast is now starting. All attendees are in listen-only mode.
Hello there, and welcome to this week's Biology 101 lecture. The topic that we will be covering this week is cells, and what we'll do is we'll spend most of our time on the first half of the chapter. The first of the two chapters. And the reason why cells are important is because in biology, it's the study of life, and really, with the possible exception of viruses, every form of life on Earth is either a cell or is composed of cells. And so, if we're talking about the study of life, studying that smallest living unit is very important. There are two basic categories of cells, prokaryotic and eukaryotic cells, and these cells have several sets of differences. The prokaryotic cells do not have a nucleus, and their DNA is spread throughout much of the cell, and that's what they're showing over here. Whereas in eukaryotic cells, there is a nucleus with the DNA contained inside. Prokaryotic cells are much smaller than eukaryotic cells. Prokaryotic cells are only parts of single-celled organisms, whereas eukaryotic cells can be part of multicellular organisms. There are single-celled eukaryotes, though, mostly protists. And there's only one type of organelle inside the prokaryotic cell, and there are many types of organelles inside the eukaryotic cell. Prokaryotic cells are composed of are bacteria, and another class of organisms called archaea. You'll talk more about the diversity of those organisms in session 9 of this class. So, single-celled. Eukaryotes contain a great many organisms, including plants, fungus, animals, and you are a eukaryote. You are composed of many eukaryotic cells. So, what we're going to do here is we're going to go through the parts of the cell, and if you notice at the top of your class session, there is a file that compares the cell to a factory. Some of the things what you're going to see today are kind of obscure concepts that you may not have encountered before. And by comparing them to the factory during this lecture, we'll hopefully be able to increase your understanding of what's going on inside the cell and what the parts are. The factory that we're going to talk about today will be a factory that produces proteins. Because really, a lot of a cell's job is to make proteins, because if you recall from last week, proteins are very diverse molecules and can do so many things, and so the cell is really going to utilize a lot of those. To do this, to make these proteins, we're going to use something called organelles. And just like that might sound, organelles, it sounds like organs. And so these are really kind of like tiny organs within a cell that carry out specialized functions to help that cell stay alive and do its job. Just like your organs help you stay alive and do your job. If I ripped out your heart, obviously you would no longer function. And if I ripped out, say, the Golgi apparatus, which we're going to learn about, from a cell, it also would no longer function. So, we have an animal cell. We're going to start off with that, and then later we will look at how animal cells compare to plant cells. But let's jump right in with the cytosol. Cytosol is basically the space inside the cell, the cytoplasm is the space inside the cell. The cytosol is the jelly that's in that space. So I've drawn a cell here. This is a cell membrane. We'll talk about that in a little bit. But this space, the cytoplasm, is like the factory floor. Right? If you're going to have a factory, it needs to have some place where it exists. And that's what we're talking about. We're just talking about the space in the cell at this point. There also is a cytoskeleton. All right. Now, the cytoskeleton functions in cell structure, cell movement, and the transport of materials in the cell. And so if you look at your factory analogy, what we call that, they say that the cytoskeleton are like the support beams that hold the factory together. And that's true. We can draw some support beams on our cell here, and these things are going to hold our cell together in some way. And that is true, but the cytoskeleton also does other things as well. You can move because you have a skeleton and your muscles pull on that skeleton to produce movement. We can do the same thing inside a cell where there can be motor elements that pull on parts of this cytoskeleton that enable the cell to move. We also can do another thing, and that's that these can actually act like hallways. Or if we want to move something around a cell, they can actually kind of use these as guides to move from place to place. So that is the cytoskeleton. It's a little bit more than just the support beams. All right, now we come to the plasma membrane. And the plasma membrane is listed on your factory analogy as the shipping and receiving department. And why would you think that a plasma membrane would be listed as a shipping and receiving department? I'm going to adjust the camera here. Well, the plasma membrane is listed as the shipping and receiving department because this is where the cell comes into contact with the outside world. This is where all the action happens. And it's important enough that your next chapter is just about the plasma membrane. And if we were thinking about a factory shipping and receiving department, you might envision that there's one entrance into this factory somewhere. However, in our cellular factory, there are a great many entrances. And so they're all over the place. Every place around the cell, there's an opportunity for things to move in and out. Obviously, as we'll discuss in the next chapter, we will selectively move things in and out. So, but if this is our shipping and receiving department, what is it going to receive? Well, it can bring in things that a cell needs, like energy. It can bring in the basic raw materials that are needed to produce something in the factory. And it needs to send out wastes. And oftentimes, it needs to send out a product that is constructed. And so let's give an example here and say we have a carbohydrate outside our cell. And this carbohydrate is a very large molecule that the cell cannot bring in on its own because it's too big. Well, in some ways, this is like getting an order in your factory because, there we go. In some ways, this is like getting an order in your factory because there are receptors on the outside of this plasma membrane, and maybe that receptor binds to this carbohydrate, and it then sends a signal to the nucleus that says, hey, there's carbohydrates out here. And remember, we can get energy by breaking down carbohydrates into simple sugars and then using those simple sugars to generate ATP. So this cell wants to get those sugars. That's the fuel that'll keep it going. And it sent the signal to the nucleus. And if you look at your factory analogy, the nucleus is called the chief executive officer. So it's like the boss. And you can almost envision the nucleus of the cell, like when you see factories and the boss sits on an upper level looking down over the factory floor. Think of it like that. Because what's going to happen is the DNA is spread around in here. But this cell just received a signal that said, hey, there's carbohydrates out here, but we can't bring them in because they're too big. So what this cell might be able to do, and something you'll learn about next week, is make an enzyme. This enzyme can potentially go in and break that carbohydrate up. And instead of being composed of, take each of those individual units of a simple sugar and split it up. Take it from a polymer of many sugars to individual monomers. Polysaccharide to monosaccharides. And so what's going to happen at this point inside the eukaryotic cell is that that DNA that's in there has important information. That DNA has the information necessary to make that enzyme. And so what this is going to do, this cell, the nucleus, is going to basically take that DNA and expose a section of it. A section of those A's, T's, G's, and C's that code for making that protein. And it will use something called messenger RNA to copy it and send the instructions for making that down to what's going to be called our assembly line. Because we want to make an enzyme to ship over here to break that up. Now keep in mind, the nucleus isn't sitting there thinking about it. It's basically the cell receiving signals and all sorts of different information, and based on that information, certain things are produced. Maybe it won't produce something because of some information. But in this case, it exposes this, sends a message to our assembly line. Our assembly line will have ribosomes on it, and these ribosomes are on what's called the endoplasmic reticulum. So, the assembly line is the endoplasmic reticulum. The ribosomes are the workers of the assembly line. And what's going to happen is this protein will start being made with one part. And then we'll add another amino acid, and another amino acid, and another amino acid, and another amino acid, until we put together a huge string of amino acids, potentially thousands. And when we've strung, and as we're adding these parts to it, you can envision it kind of moving down the assembly line until it gets to the end. But when it gets to the end, is that the end of the story? We've used the mRNA, our ribosomes have put it together, but we need to do something with our product, right? Now imagine you ordered something from Amazon. How are they going to get it to you? Well, they're going to put it in a box to keep it safe, and they're going to slap an address label on there so it knows where to go. And that's what happens at the end of our assembly line. We use something known as the Golgi apparatus, which we can think of like a finishing or packaging shipping department. And what we do is we put a vesicle around this enzyme that we made. And in this case, we would then shift or send this vesicle up over here, excrete it outside, which we'll learn about in the next part, and then the enzyme could start doing its job of breaking up our simple sugars, our carbohydrate into simple sugars. And of course, those are the main parts of the factory. We talk about making a protein, but let's look at some other parts that are involved in this factory. Lysomes and cellular recycling. Lysomes are organelles that break down worn-out cellular structures or foreign materials. And your factory analogy says that lysomes are like the maintenance workers. And that's true, because one of the jobs of the lysomes inside the cell is, say there's something up here that's broken, the lysomes will go up here, fuse with it, digest it into little tiny parts. And those little tiny parts will follow one of two paths after it's broken down. First thing they can do is excrete them because they're wastes. There's nothing to do with them. But our factory is also environmentally conscious, because when it breaks down one of its own parts, if there's something left over that's still usable, it recycles it. So say we broke down a protein in here and we had some leftover amino acids, we would keep those amino acids and send them to the assembly line so they could be used to build something new. Last week we talked about how proteins were like putting together 20 different Legos. You're not just going to destroy something with your Lego. You're not just going to build something with your Legos, take it apart, and throw those Legos away. You're going to reuse them to build something else. And so that's their job as maintenance workers. But lysomes can also function as security guards, because in addition to breaking down things that are part of the cell, they also break down foreign invaders. So if something gets into the cell that the cell does not want there, the lysomes will go and attack it. Basically use those same digestive enzymes that they use to break down themselves to break down foreign materials. And so there we see an example of a lysosome fusing with something to break it down. Mitochondria. So we have our lysosome over here. There's lots of lysosomes all over. Mitochondria inside the cell, in your factory analogy, are listed as the power plant. All right, think about it. A factory needs energy to run, just like a cell needs energy to run. And if we're in Indiana, and we have a factory in Indiana, our power is coming from coal. Most likely. We have other sources, but we're predominantly powered by coal in Indiana. And the way we're using that coal is we're breaking bonds inside the coal to go through a process that ultimately turns it into electricity. And we send that electricity to the factory. And that factory can run and do everything it needs because it's all powered by electricity, right? Electricity is a pretty good kind of universal power source for most things that you're going to do. And that's what it turns out the mitochondria do inside the cell. They take sugars from the cell, a form of chemical energy, and they turn them into ATP, which can be used to fuel a whole bunch of different things. And so just like the coal, we take chemical energy and turn it into electricity. Here, we're taking chemical energy and turning it into ATP. And then that ATP can be used to do anything in the factory. Mitochondria are kind of cool because they have their own DNA, and they are passed down through the maternal line, not the paternal, because whatever DNA are in your mom's egg, they move forward. And so there's a lot of unique characteristics. Because they have their own DNA, you can kind of see the origin of eukaryotic cells, where this is almost like another little cell living inside that one. So we have our power plant from our mitochondria, and here they are. We already kind of talked about our cytoskeleton, so we're not going to do that again. Basically, remember, it's like a skeleton, but it also can be pulled on to move, and it can also act like hallways. There are cellular extensions on the outside of cells, so it's tough to kind of use the factory analogy for this one, but cilia may extend from a cell, and these can do a lot of different things. They can help, for instance, spread around substances. You end up with, in your throat, places where mucus is produced. But we need that mucus to evenly cover the surface of your throat to provide that protection. And so you have cilia on top of those cells wiggling around like that, spreading around the mucus all the time. Another type of cellular extension is a flagella. The only place we really see flagellas are in single cells where we need to move that single cell. And so, for instance, there's only one type of flagellated cell, or cell with a flagella, in humans, and that's in sperm. Let's talk about a few things that are cool about plant cells. Plant cells have everything that animal cells do, but they get a few bonuses. These include a cell wall, a large central vacuole, and organelles known as chloroplasts. And so here's a picture of our cell. First thing is, the cell wall, you can think of it kind of like a fence around our factory. Because the cell wall lies outside the plasma membrane, and it's an extra layer that plant cells have that animals don't. And you'll see when we get to the section on plants that the cells in plants orient like bricks. And they can do this because they're not mushy like animal cells. They've got this rigid cell wall that helps them keep their structure. The next thing we have in that list was let me just double check it, the large central vacuole. And what we do with the vacuole is we store nutrients and water and things inside. And so we think about our factory analogy. A large vacuole would be kind of like a storehouse, where we're going to store things that we need to save for later. In animal cells, there's not one big storage area. There's a lot of little ones spread out. But in a plant cell, you can have this very obvious vacuole that is filled with things. So here's our vacuole here. And now we get to our last one. And this one's kind of interesting because it's the chloroplasts. Okay, so we know that plants are green because they use photosynthesis, and so they have chloroplasts, right? And your whole life you've learned that plants get energy from photosynthesis, animals get energy from eating things like plants. And so plants should have chloroplasts, animals have mitochondria. However, here's the thing. As part of that story, many people end up thinking that plants have just chloroplasts and animals have just mitochondria. But if you look at our picture of the plant cell here, you will see that, in fact, we have drawn mitochondria inside our plant cell. And here's the really important point. When we talked about photosynthesis last week, we showed you thing with the sun. We're going to take the carbons from the atmosphere, put them together to make sugars. We've made a sugar. And we've taken energy from the sun and stored it in chemical bonds inside our sugar. Well, how do we use that energy that's in that sugar? We've made a sugar. The end of that story inside the plant wasn't that, wow, magically the plant has energy. It's got to use that sugar. And so what plants are actually doing is using photosynthesis to build the sugars, but then they also need mitochondria to break down those sugars to generate their own ATP. So a plant cell will have both mitochondria and chloroplasts, whereas an animal cell just needs that mitochondria. The animal cell is not a primary producer. It does not make its own sugars. It eats sugars that other things have made. And keep in mind, I'm using sugars in general in a term for biological molecules that can yield energy from plants. All right. So those were our differences between the plant and the animal cell. And you've got a handy chart in your book that highlights some of these things. And you can compare that chart to our factory analogy, where you can use it to basically kind of as a little study guide. So let's go ahead and move on to the next chapter on the plasma membrane. The plasma membrane is a thin, fluid-filled entity that manages to be very flexible and yet is stable enough to stay together despite being continually remade due to the constant movement of materials in and out of it. And so really what we're talking about with the plasma membrane is a membrane that is composed of these molecules that have polar heads and nonpolar tails. And so this ends up being, the consequence of this is the phospholipid bilayer structure of a membrane. And what do we mean by that? Well, let's go ahead and we'll draw what our membrane would like zoomed in over here. You have molecules that are fats. Remember, oil and water don't mix, so lipids and water don't mix. And these are lipids. They're a type of fat that's shaped like this. And in this fat, these heads really like water, and these tails do not like water. So if you just plopped one molecule of this into water, it would not be very happy. The head would be like, woohoo, water everywhere, and the tail would be sad because it would have to be near water. It's repelled by it. And so what happens is, when we make our membrane, is we can use these molecules in a very interesting way. What we can do is we can string together a bunch of these molecules to make a layer. And if I made this layer and put it in water, it still would not be very happy because these tails would all still be exposed. But if I take a second layer and go like this, what I have now done is isolated these tails in the middle. And if you think of this like the outside and inside of a cell, water outside the cell, these guys are all happy. Their polar sides are touching water. Inside the cell, these guys are touching the cytosol, and so they're happy. And these middle tails are not touching any water, and they're happy. And this is where we get our phospholipid bilayer from. And this phospholipid bilayer is not just the lipids. There's also proteins in there. And we'll look at what those proteins that are integrated do in a moment. One last thing, this is a fluid layer. And what we mean by fluid mosaic is that it's constantly shifting and moving. One individual lipid molecule that starts here may end up a ways away on the cell later in time. You can think of it like imagine there was a pool. And you went to that pool and you threw in 100 white beach balls and 10 red ones. and you took a picture of it before you left. You leave, you come back later in the day, end of the day, and you take another picture of the pool. Will those balls be in the same positions? No. During the day, wind and movement will have shifted them around all over the place. The surface of the pool would still be covered with the beach balls, but nothing would be in the same position. And that's what we mean by the fluid mosaic model. So we're not going to... All right, membrane protein functions. Proteins can do a bunch of different things inside membranes. Some of them provide structural support. So, for instance, in this case, we have a protein that's attached to the cytoskeleton, right? So remember how we said we could move things around or we could hold the cell together? Well, those support beams and walls have to touch this outer cellular membrane. One other thing that you can do is provide sites of recognition. So on this membrane, there are markers that identify the cell. And this is important in the example your book gives you is for your immune system. Your immune system has white blood cells. Those white blood cells are cruising around looking for bad guys inside your body. But the white blood cells really ask two questions. They first ask, are you part of me? Yes or no? Like, are you part of this, your individual, your person cells? You have a bunch of cells. They all have your markers. So are you part of me? And the second thing they ask is, well, if not, who are you? So that I can best destroy you. And so with that first question, it's pretty important because it means your immune system, if it does not identify a cell as being part of your body, it will try to destroy it. And this is why we have problems with organ transplants. Because what happens is your immune system finds that organ that was part of another person and it says, this is something foreign, I'm going to kill it. And that's why in organ transplants we have to find close matches, because the closer your cellular marker proteins are to somebody else's, means the less likely your immune system will fight it strongly. We also have proteins for communication. I already showed you an example of a receptor that might bind to carbohydrates earlier. You can also send signals out. So for instance, if we were this cell that was a factory and we were producing a hormone, right? It's got to get outside the cell somehow as well. And so a cell could communicate with proteins as well. So it can, let me rephrase that. It can both receive signals from hormones, although we would create them inside the cell. I misspoke there. Proteins can serve as channels that things can pass through. And we'll talk about that in just a moment. Okay, so let's talk about diffusion. Diffusion is the movement of molecules from a region of higher concentration to a region of lower concentration. And the best way I can describe this to you is to think about a time when you were driving down the highway and you smelled a restaurant. Let's use McDonald's as an example. So you are cruising down the highway and you start to smell McDonald's a little bit. As you get closer to the McDonald's, what happens? Well, you start to smell it more and more. And then after you pass the McDonald's, you start to smell it less and less. What's happening is a process of diffusion. McDonald's has its McDonald's kitchens, and they vent them out into the air. And what's happening when they vent that out into the air is there's a very high concentration of the molecules right here. And so when you're right next to the McDonald's, you smell it more. But those molecules then spread out from that source. And the reason they spread out is because molecules want to spread out evenly in their environment. They want to spread out to fill the space. And so you can think of this in terms of the example your book gives you, which is this putting a drop of dye in water. You start out with dye molecules, they're clumped, it's very red, but then those dye molecules will start to spread out evenly in the space. So in this case, we're talking about a solvent, which is water, with a solute, which is the dye, and that solute wants to spread out evenly or as much as it can inside that water. And so what this means for a cell is it can take advantage of this to sometimes move things around. So let's use our cell here and kill some of our organelles inside, just so that we can show what's going on. If we have our cell, and let's say that it wanted to get those sugars in that we've broken down. Or actually, let's use an example of salt. Let's say it needs salt. And because we'll talk about sugars in a different way in a minute. And there is salt outside the cell, and there's very little salt inside the cell. What's going to happen is this salt wants to spread out as evenly as it can, just like that dye did. And so you will have the salt move into the cell such that there's an equal concentration on both sides of the membrane. And so that's great for when we want to get to move things from an area of higher concentration to lower concentration until they equal out. Also, let's say we had some waste that we didn't want. As long as there's more waste on the inside than the outside, that waste can sometimes move out of the cell due to diffusion. So diffusion is a nice way to do things because it does not require the cell to use any energy. It takes advantage of this natural property of chemicals to want to spread out evenly. The membrane is semi-permeable. And what that means is it's going to let some things out and in selectively. It's a selective membrane. If everything could just move in and out as it wanted through diffusion, what would be the point of having that membrane, right? It's part of one of the requirements for life is to, keep yourself together and organized. And to do that, to maintain homeostasis, it's going to need to regulate itself. And it does that with the plasma membrane. So smaller substances can sneak through that membrane sometime. They're small enough they get right through. Other substances may be too big to get through that membrane, but they could still diffuse. And so, for instance, here we have an example of those sugars that we made earlier. They cannot just sneak through the membrane. But what we do to help with that is we create what's called a transport protein. And in this case, the transport protein is going to be involved in facilitated diffusion. And in facilitated diffusion, we basically made a tunnel through our membrane for molecules to pass. And so this works out very well when there's a high concentration outside and a low concentration inside, because those want to diffuse down their concentration gradient. But sometimes we have a problem. What if we had a whole bunch of, let's say, sugar inside the cell, because we've stored it up, or we could say salt, whatever, a whole bunch of something we want, but there's less outside. In this case, diffusion would tell these molecules that we wanted to move outside the cell. That's exactly what we want to happen. We want to bring more of them in. These are useful resources for us. And so, in this case, what's going to happen is this cell is going to have to move these molecules against their concentration gradient. And in order to move something against its concentration gradient, to fight that process of diffusion, you have to use what's known as active transport. And in active transport, we use that ATP that we talked about earlier, what we're generating from our mitochondria, to move these things against our concentration gradient. And so a lot of these are known as pumps. And so in this case of a glucose molecule, so those sugars we were talking about before that we want to move against our concentration gradient, they bind to a receptor on the outside of this. They bind to a receptor. That receptor says, hey, there's a sugar here. Let's let it in, and it lets it in. And the movement of these, if you imagine that, like, here's a cell, and my arms are the protein in the middle. We start off with our arms like this, and here comes a molecule. It binds there. Then to let it in, all we do, change the shape, and it's been let in. And so what's really cool about this is a lot of times these move like this, and when it lets something in doing that, something else from the cell will bind on the inside and it will let it out. And you'll see a little bit of that in anatomy and physiology, but not much later in the semester. So we can do that with pretty big molecules, but what happens when molecules get really big? Well, when molecules get really big, we have to use processes known as exocytosis and endocytosis. And what goes on with these processes is we're going to use those vesicles that we talked about earlier. And so let's say we had produced something that we wanted to get outside the cell that was really big. We wrap it in a membrane. We then send that membrane, and the outer edge of our vesicle fuses with the inner edge of the cell's membrane. And so we've got our substance in here. Once those two things have fused, we can then degrade part of this, and this can sneak out. But notice what we've done. We've almost created like an airlock, because things still can't just flood inside the cell, because there's still this membrane holding it in place. And so all we have to do is fuse, dissolve part of it, let it sneak out, and then eventually what we'll do is, because we don't want a lumpy membrane, is this will eventually subside back to the resting state of the membrane. And so that one's pretty easy, but sometimes we also have to get really big things inside our cell. We need to use endocytosis. And in endocytosis, what we're going to do is try to bring something in. And we are going to do this with pseudopodia, which are basically false feet. I pronounced that wrong. Don't worry about it. In this case, let's say something that looks really good lands right here. And we want to bring that in. And so what we're going to do is use our membrane again. In this case, we're going to create an extension of our membrane like that. So this is intracellular space. Now this extension has intracellular space. And if we do that on each side, or surrounding, I should say, a molecule, we can then degrade this and the molecule gets inside without letting the outside world in or the inside world out, because the membrane is still contiguous. And so then this molecule comes in, this is empty, we reabsorb these, bring our membrane back to normal. And so that brings us to pretty much the end of our time for today. But I wanted to make one last point to reiterate how important cells are to life. If you've ever thought about your metabolism is how much energy you're using in a day. How much energy you, as a large multicellular eukaryotic organism, is using is a function of the energy used by every individual one of your cells. Keeping you alive means keeping each of your individual cells alive such that they can do all the processes that we talked about today. An example of where that fails is in, if you've heard of bed sores. Bed sores are caused by a lack of blood flow to a region of your tissue. What happens when there's a lack of blood flow, those cells are unable to get adequate oxygen and adequate nutrients. When they can't get adequate oxygen and nutrients, those individual cells all start to die. Because we need to keep all of your cells alive. And as such, these processes are constantly going on, billions of times per day. We're making proteins, moving around, doing stuff inside your body. That's use a large multicellular organism. In session 9, we'll also look at the importance of a lot of single-celled organisms as well. And so with that, we will end our presentation for today, and I hope to see you here next week.